Hello, everyone. Welcome to the NIH's Wednesday afternoon lecture series. It's my pleasure today to introduce our Wallace speaker, Dr. George Church. Normally, this would be done by someone like Dr. Collins, but he's otherwise occupied today and couldn't attend, so I'll be introducing Dr. Church. If anyone's looked at his resume, it's very impressive. Because I'm sure you'd rather hear what he's going to talk about than hear me talk about him, I'm going to keep my comments very brief. He went to Duke University, where he received degrees in chemistry and zoology, and then went on to get his PhD at Harvard in biochemistry and molecular biology. He eventually made his way back to Harvard, where he is currently a professor of genetics and the director of the Center of Computational Genetics. While he was on his educational path, he had a number of advances related to computational biology and computational genetics, including the development of crystallographic software that led to the first high-resolution RNA structure and the development of the first automated DNA sequencing. He also helped develop the first direct genome sequencing methods. He's been instrumental in various aspects of the Human Genome Project and in the use of molecular multiplexing for high throughput biological assays. He's been a major player in the field of synthetic biology and in the push for personal genomics. He has a huge list of publications, very high-quality publications, and the most recent ones, I think, reflect his focus on using genomic applications to create high-throughput technology to make personal genomics a reality, and the use of personal genomics in medical advancement. I suspect that's what he's going to be talking about today, and so with no further ado, Dr. Church.
Thank you very much for that introduction and for everybody here and some old friends I've seen already. This is my thank you slide. It also acknowledges relationships that I have with some government and commercial collaborators of various sorts and funding agencies. So I'm very grateful to these, and I'm grateful to graduate students and postdocs and collaborators I'll mention as we go along here. The long title was really just kind of to emphasize that we're trying to develop basic enabling technology that we think might already or someday impact all of the national institutes, and they have impacted us in various ways as well, very positively, and so thank you to them as well. I also want to get out of the way, another embarrassing fact, which we still haven't sequenced any human genomes in their entirety. There's still about 5% missing, and it's not a trivial 5% either, or at least it may not be, in that here's an example of a association with multiple sclerosis that's sitting in one of the biggest gaps in the genome, one of the 300 gaps, and this one has this potentially very significant genetic association. Now, I don't think I need to tell you that we have recently brought down the cost of sequencing by about 1 million-fold, and the comment back is they'd like some more. We'd like some more. Thank you. And so how did we do this? How do we have the digestive capabilities to, and exactly what are we doing with this that's useful? And this comes down to the obvious inherited genetic tests, which build on a strong history in medical genetics. There's new methods that are much more feasible now, which looks for de novo mutations, differences between child and the parents. And this allows us to extend medical genetics beyond cases where the usual case where you have require medical history, which may not happen in the case of adoption and new combinations where you're the first person in your family. And this is a very important extension of medical genetics. And then, again, the more obvious oncogenomics, where, in a certain sense, personalized genomic-based medicine is already practiced, and pharmacogenomics that extends beyond cancer chemotherapies of various sorts. We can now do structural chromosome variations, we can do quantitation of RNA, chromatin, methylation of DNA and proteins in chromatin. Microbial and immune system, we'll talk about many of these things in a moment. We can do in situ sequencing. We can look for very, very rare cells. And in a certain sense, there's this feeling that we're replacing the high-throughput chips pathology and cytology practice that has been in place for quite a while. Now, as an example of this, here I've chosen 4 articles. I could have chosen many others, including some intramural success here at NIH, where cohorts as small as 1, in the range of 1 to 20, not the gigantic cohorts that we've come to think about for modern genetics, but these small cohorts are yielding very exciting, very medically causative and significant alleles, with as little as 10 genomes producing 22 alleles, including confirmatory sequencing, very targeted at individual alleles or genes. And so, for example, and by genomes here, I mean either genomes or exomes. I'll use them somewhat interchangeably. You can see the examples here. Jim Lupsky, who here is both first author and research subject for a neuropathy that was published. And here, Heather and Logan Madsen are adult children's in a family where all 4 members were sequenced, and that led to not 1 new allele but 4 new alleles, 2 compound heterozygotes producing 2 different syndromic. They are unfortunate enough to actually have 2 recessive combinations for 2 different diseases. And the list goes on. Some of these were done by Jay Shunduri, who was a graduate student, MD and postdoc in my laboratory, and is now a professor in Seattle. We have the cost now for an exome, which is about 1% of the genome, is around $4,000, and the price for a genome is around $7,000. And the distance between research and practice is getting very, very small, perhaps close to 0. In this anecdote, and it is just an anecdote, reported in Nature not too long ago, where a child of the age of 3 had more than 100 separate surgeries already before his 4th birthday, and obviously diagnosis was not going well, perfectly, and so they, in desperation, did sequencing, and this changed their viewpoint from it being an intestinal problem with these multiple intestinal lesions to something that was immune system dysfunction due to apoptotic gene XIAP, and so they changed to a bone marrow transplant, which had impact within a couple of weeks, and the child has been fine ever since. Now, generalizing this beyond anecdotes and focusing on medical genetics, not direct-to-consumer genetics, which I won't talk about, but real medical genetics as commonly practiced, there are now over 2,200 genes which are considered highly predictive and medically actionable. It may not be the perfect cure, but there are medical devices, treatments, therapies of various sorts for these 2,200 genes, and it's better to know what it is you're dealing with than merely knowing the symptoms. An early success with phenylketonuria is one of about 40 genes which are routinely analyzed, usually by enzyme assays from a heel prick of blood from 4 million infants per year in the United States alone. And then one of the first and foremost of the 2 genes, BRCA1 and 2, in the early 90s established as a DNA sequencing-based test, and these have been extremely heartily embraced by the community, and they have been continued to increase quite steadily during the era of the Genome Project and beyond to now 2,200 plus genes. Now this does not, even though those are highly predictive and highly medically actionable, it doesn't mean that everything about genomics is, and it doesn't mean that our failures to have, in cases where biology is less predictive, it doesn't really count as a failure. It's a work in progress, and there are plenty of things that work. And we're not saying that we're going directly from your personal genome with 3 million differences between you and the reference sequence directly to traits. That's not the idea. The idea is environments is a big component, and the revolution in sequencing is partially spilling over into environmental measures, as you'll see momentarily. Plus all the electronic sophistication that we have is beginning to have impact in terms of standardization and recording of environmental components. I'm going to mention this sort of in the context of the Personal Genome Project, which started kind of accidentally in an ELSI component, a one-page ELSI component of a 2003 grant to NIH, NHGRI. But it now includes centers in 4 cities and 3 countries, and the idea is to measure these environmental components or capture them from medical records, electronic medical records, personally controlled health records. Measure immunome and microbiome, we'll get to that. And measure the intermediates to the extent possible of going through epigenetics, RNA methylation, and so forth, either directly from cell samples or via reprogrammed stem cells. And I'll show you that. Now, this project is based entirely on individuals willing to have their genomic data, their cell data, which includes saliva, blood, skin, and induced pluripotent stem cells, and extensive trait data, some of which I've mentioned, in an open access, Creative Commons 0 format. This is as open as you can get, really. And there are now over 13,000 volunteers that are registered internationally. Over 2,000 of them have achieved 100% on entrance exam. This is a deviation from the standard practice of giving a consenting, 5 to 20-page consent form that most people don't read, and most principal investigators are aware that they are not reading them. And we have over 1,000 of them have their medical records online, and 66 countries. Now, this is really meant to be mostly an icon for how important it is to embrace our outliers, not just to look at in the middle, but to look at the high deviance. Now, this is not intraspecies, but interspecies deviation. But it's to say we want to embrace these outliers in a variety of ways. And this happens to be a database, an aging database that Pedro de Magalis put together over the years, many of them in my laboratory. And we are in the process of sequencing some of these outliers that have, for example, naked mole rat, which lives about 10 times longer than its close rodent cousins, and capuchin monkey. The bowhead whale lives up to 200 plus years, and then this clam lives over 400 years. And there are many humans. Here's a human celebrating her 121st birthday, and I sincerely hope that many of us in this room can do likewise. But right now, these are outliers. The oldest, and we intend to get, we're looking very much at older families, family members that have, and this is our oldest complete genome sequence so far in the Personal Genome Project, and as far as I know, internationally. He's of African-American ancestry, and so he has sickle cell trait. Obviously hasn't put too much of a crimp in his lifespan, even though it is one of the few things that the U.S. military actually diagnoses because of presumably high altitude implications at one point they felt was of significance. And I want to point out this resource, evidence.personalgenomes.org, where we're actually building community resources for annotation of this connection between genes, environments, and traits, both automatic and manual curation, to make it so that you can go from, as well as you can, from genes and environment to traits in a way that the community can critique rather than some proprietary system which is more difficult to critique. So this is sort of a Wikipedia type of model, but with slightly higher accountability for the individuals editing it. And part of the motivation for that kind of personal report was when you put data in a public domain, then it's possible, in fact likely, that the individuals will get access to their own data, and then you have the choice of letting them go off and interpreting their data with their own computer programs or some fly-by-night operation that they find on the internet, or we can build a community where we can critique the process by which people analyze their full genome sequences or partial genome sequences. And here's an example of one of the gray zone. This is not our easiest case. This is one of our most difficult cases, where PGP number 6 was found to have a particular allele, an alanine 13T in the MYL2 gene, which was known to be one of many genes involved in hypertrophic cardiomyopathy, many of which are very well established, many alleles and many genes are very well established for this disease. And it is a very important and interesting diagnostic situation, because this is not part of standard medical care. You don't normally get diagnosed for hypertrophic cardiomyopathy or tested for it unless you have family history. But it is something that can act very swiftly, rather than the kind of long-term warning that you might get for atherosclerosis, where you might get tired going upstairs. This can hit even athletes very suddenly and cause death without a lot of warning. Anyway, we felt that there was only one case in the literature of this exact allele in this gene, and we felt that this was really insufficient to get too worked up about it, even though this person had the same allele. We wanted to avoid the incidentalome, but we also felt, at least the physicians that are part of the Personal Genome Project felt, that if we asked around, we would find a lot of additional data hiding in people's filing cabinets, companies and clinics and individual investigators. And sure enough, we did ask around and did find many more cases of this A13T allele, many of which were affected with hypertrophic cardiomyopathy at relatively early onset. And so we felt that this wasn't perfect, but it was enough to recommend to this individual to get an echocardiogram looking for an increased thickening of the ventricular wall, and since it's uncertain when the age of onset would be, to do that every 2 years, and that's what he did. This is kind of a dry slide, but it's to tell you that there are many uses of the Personal Genome Project data as well as tissues already. These are published in decent journals, ranging from sequencing itself to analysis of methylation and microbial diversity, hypermutability, and stem cells. Now, why is it that we can do all these applications, that we can sequence all these individuals and animals suddenly? And part of it is kind of a standard Moore's Law curve, where we get a factor of 1.5-fold. It's an exponential, as actually a pretty fast one, ever since I was a teenager, basically, for computers and for DNA sequencing. But then what happened is sort of about the time the Genome Project was being declared a victory, and it changed its slope to an even steeper exponential of about factors of 10 per year, resulting in about this millionfold effect. I don't think we're going to get a factor of 10 this year, unfortunately, but it's still going to be quite striking exponential for a while longer. And we're in this sort of this range of $1,000 cost to around $7,000 price, meaning that's what you would actually have to pay in fairly large bulk projects. And the reason that this has happened in part is because of breaking of a monopoly that existed during the Genome project, which I think many of us found quite frustrating. And maybe we've overcompensated by now having 22 different technologies, all of them different from the one that produced the draft sequence. And my group and I are involved in almost all of these, about 18 of them. And in the little box here are the 6 that have been on the market available for about 2 years, at least 2 years now, some of them longer. And there are a couple more coming soon that I'll mention. And I don't necessarily see this as a strict Darwinian situation where there's only one winner. I think there are these 7 different niches, you know, the cheapest per base pair, the cheapest per run, the least expensive instrument, long reads, dealing with repeats, speed, portability, and so forth. I think these niches may be dominated by one machine, or they may be 7 different, or they may be a variety. But I'm going to walk you through 3 very briefly just to give you a feeling for how it is we go about thinking about inventing 20 new technologies and optimizing them and bringing them into the market. And this is one that actually represents quite a few of them that were in that box. It's a fluorescence bait. Many of them are fluorescence based. They involve small amplifications or single molecules with sequencing by synthesis, which can either be sequencing by ligase or sequencing by polymerase. And it has 2 key components. One is placing single molecules or slightly amplified from a single molecule either in a random grid or in this case actually an ordered grid. This is a complete genomics. And then for each of these single molecules, which contains synthetic DNA in yellow and unknown genomic DNA in green, is extending from the known regions into the unknown with either polymerase or ligase with fluorescently labeled 4 colors, A, C, G, and T, representing typically about 1 base at a time, but billions of these simultaneously. So you can have, it's fairly typical to have a billion of these reads going on simultaneously in a run. So this is now a beautiful interaction between single molecules and microfabrication, and the single molecules are amplified just enough so that they now completely cover this 200 to 300 nanometer microfabricated dot of positive charge, and we call this the Polyny exclusion principle. For the physicists in the audience, this is intended so that one molecule will bind there, and you get the moral equivalent of clonality without actually doing standard cloning. And this just shows how that grid is really clonal when we do one round of sequencing where we get A, C's, G's, and T's, the 4 colors here, and you can see these are very clean. You have very little overlap of colors, as you can see more quantitatively here. And you also see the difference between this grid, in this square grid, and the random distribution that we had in the early days in the late 90s and early 2000s, where we would randomly distribute these polonies or clusters or single molecules. And they get about a factor of 5 or so due to the difference in the Poisson distribution where all these black regions are not genuinely useful. Plus, we've now improved the density. Because it's a square grid, you can actually get alignment with the digital camera, and you can get another factor. You can, instead of getting 25 pixels per base pair, you can get as little as 1 or 2 pixels per template. And that's another factor of 10 or more. So that's one example, and this kind of characteristic of all the ones that have been available for the last 2 years is, I'm going to show you 2 examples which are not quite as reduced to practice as that. This is ion Torrent, which is a variation on the pyrosequencing method, where where you're detecting this reaction of multiple molecules. This is not a single molecule method, where the triphosphates react with a primer, here represented by the OH, 3' OH, to release pyrophosphate and protons in this reaction. In the 454 Roche method, you detect the pyrophosphate by a coupled set of enzyme reactions that result in bioluminescence. And in this, you detect the proton directly through a field effect transistor array. But in both cases, whether it's a camera or a field effect transistor array, you basically have a microfabricated chip that either detects protons or photons. The thing in common with the 454 and this ion torrent is that when you have a run of bases, you typically have to measure the height, the integrated intensity of the photons or the protons coming through here. And that can lead to some ambiguity when you have very long homopolymer runs. But this is probably the most miniature and the least expensive starting instrument. And we'll come back to that in a moment. This is even further in the future. It's also pretty far in the past. This is one of the first sequencing methods that my laboratory worked on in collaboration with Dan Branton and others. And it was really beautifully reduced partially to practice here by Hagen Bailey and his coworkers, where they showed this is actual picoampere current going through one of these highly modified hemolysine protein single molecules, where you can see the effect of putting in A's, G's, or T's as monomers through this pore blocks the current going through it by discrete amounts, here color-coded with C as blue, and and you can see it dropping down into the blues, whatever you throw a C through, and there's C's, G's, T's, and A's having discrete levels, which are represented in this histogram here, where you can see very little overlap in the characteristic pore current for detecting these mononucleotides. There's a variation on this which uses polynucleotides, which is sensitive to mononucleotides, and it's an open question as to which is going to win, the mononucleotides embedded in polymer or broken free. But the point is, this is not complete pie in the sky, and it's quite exciting. Now, how do we evaluate this? One of the things that we need to evaluate is errors in throughput. So just to put this in perspective, these two sequencing by ligation methods, the complete genomics methods and the applied biosystems latest model, has on the order of 1 billion reads per instrument. And the two that are just coming around the corner right now are, in a way, much lower number of template molecules, but they're reading a bit faster. So these are not in the billions, but in the hundreds of thousands, about 100,000 or less. And they have a much higher error rate, as high as 15% relative to the 0.1% that you can get from the dual nucleotide encoding that you get in sequencing by ligation. And here's just an example showing in the applied biosystem system how that you can get a noticeable decrease in the raw data quality. Here's for the 2-base encoding that they typically use, where you have, this is around 10 to the minus 3 raw error rate. And of course that can, with consensus, all of these can be improved sort of towards the 10 to the minus 6 error rate that people come to expect now. Now that's all very cool technology, but, and the big but is that if you have a phone, whether it's a landline or a cell phone or a computer network, and you're the only one on it, that's not really great. And the question is, how much sharing do we need in order to really get the awesome power of genes, environments, and traits? And as I've said, I'm inspired by Wikipedia, which has what one would think is one of the worst possible editorial policies, where you let anonymous individuals be editors worldwide, but it is really, I think, a remarkable resource, and it's something that we should study. And so you can ask, who should have access to the genes, trace, environment data? Should it be only the experts, the ones that wrote the grant and collected the data, and their close colleagues? And I would say we don't know who is going to make the breakthroughs. It could be, real outsiders and here's some of my heroes that really are only insiders in the sense that their family is affected by these various diseases. Here's one that was immortalized by Hollywood in Lorenzo's oil, and this is the real Augusto Odoni here with his son. And this is Hugh Reinhoff, who just told me in an email recently that he's figured out what his daughter has. It was an unknown disease, but again, he's doing this more as an individual than as a member of an insider team. Kay All was an undergraduate in one of my classes, and after she was out of the class, she went off and started studying her own genomics because of hemochromatosis that was in her family. And Jamie Haywood has this website, Patients Like Me, where people actually reveal quite a bit about their neuropsychiatric, their sexually transmitted diseases, and a number of things that you would consider quite stigmatizing, but they're quite open about it, and they find value, both medical, scientific, and social, from this exercise. Now, we have a big, in our project, and indeed in many other projects, this emphasis on environmental, and the idea of genetics is in part to help us stratify and segment diseases that look like they're homogeneous, and that helps us understand the environment. And the idea is, we're not saying, here's your genome, your genomic destiny, get used to it. It's more a matter of how can we find people who have either already, or how can we avoided their genetic destiny by changing their environment. And here's an example of PKU, which is nearly 100% predictive, and you could consider it 100% genetic. Prior to being able to diagnose it, it would have resulted in mental retardation with close to 100% certainty. But now it is 100% environmental, because you can avoid dietary phenylalanine, not a trivial exercise, but you can do it for your children if they are homozygous for this. And BRCA1 and 2, that I've already mentioned, the common medical action is bilateral mastectomy and ovarectomy, which you clearly don't want to take lightly, but it is very common action and considered quite important. Now, that is a pre-existing predisposition to cancer, is part of it, the inherited part of cancer. In addition, there's a somatic component that I don't need to tell this audience about, that a multi-step process by which you go from possibly some inherited predisposition plus some possible environmental components, mutagenesis and so forth. And then finally, you can get a set of mutations that once you undergo chemotherapy or some other cancer therapy, become resistant. And here's an example of something I think is quite important going forward, is our ability to detect rare cells. So in green are cells that we can genomically determine in a certain sense in situ, in that each of those green dots represents a single cell, and we're analyzing it. It certainly is the combination of inherited, somatic, and cancer-causing mutations, and mutations where we know the impact on the binding of a drug molecule, where these mutations, the three-dimensional structure is known, and the mechanism by which you get drug resistance is known. So you can track this at the single cell level, and the idea would be to detect this as early as possible and as rare a cell as possible, as long as you're not creating false positives in the process. So that's an example of where we'd like to go with sequencing, which is towards in situ sequencing, and we'll come back to that in just a moment. It also illustrates this issue of drug resistance, which can be due to these point mutations in that case in the treatment of BCR-ABL kinase, and in this case multi-drug resistance and extreme drug resistance that occurs in a major global pathogen, tuberculosis. This was done in collaboration with the Murray Lab, the Broad Institute, and Mike Strong was a postdoc in my lab that did this, where the full genome sequence of many strains that were extreme drug resistance and multi-drug resistance were compared, and individual point mutations, and this is can be fit onto the three-dimensional structures or homologous three-dimensional structures of such targets as gyrase and RNA polymerase and so forth. And each of these can potentially suggest the mechanism by which these point mutations cause drug resistance, and possibly new classes of drugs that could be used in combination to nip off this multi-drug resistance issue. Now, this illustrates our particular view on microbiomics, where we are not sequencing the entire genome of all the microorganisms, fungal, bacterial, viral, and so forth, that exist in abundance in every human being, but we're targeting a tiny fraction of it, not even smaller than the fraction that we targeted in exome sequencing. This is 0.01% or so, where we're specifically looking at drug resistance and pathogenicity alleles. This, not just genes, but it could be as little as a base pair. And so sometimes, or very often, it's just knowing what organism you're dealing with isn't sufficient. You need to know how many antibiotics. And we'd like to make this high throughput as possible. Now, in the process of doing this, it's studying Personal Genome Project volunteers over the course of a long time series, over the course of a year or so, and looking at environmental samples. Gautam Dantas, who's now at Washington University, and Morten Sommer, who's in Copenhagen as a professor, stumbled upon this phenomenon that we published, where that we found bacteria, multiple different phyla, almost every major phyla, could be found to have members that could not only be resistant to 50 times normal clinical doses, not just to one antibiotic, but to all 18 classes of antibiotics that we tested, and not only resistant to them at these high levels, but actually subsisting on them as their sole source of carbon. And so that, this is kind of an interesting anecdote, but it illustrates the more general topic, that is that we want to be able to monitor in a routine manner subsets of the microbiome that we think are medically relevant, so that we're not surprised, and this can be integrated with other aspects of epidemiology or Personal Genome Project. Now, it also illustrates that, okay, doing direct microbial genomics is important in cases where you're in a big rush, or you're looking at drug resistance. But the alternative is looking at your immune response to microorganisms, and this is, commonly practiced in diagnostics, like for HIV and tuberculosis. Typically, that immune response won't tell you whether you had an immune response to a drug-resistant or a multi-drug resistant organism. So these are very complementary approaches. But I want to just pursue for a few slides here how we would generalize these single-purpose immune tests, which have been of great diagnostic significance. So how would we generalize the immune part of this microbiome immunome axis? And so this is a project that would have been ridiculous a few years ago, but because sequencing is 1,000,000 times cheaper, it's only mildly ridiculous, which is sequencing the entirety of the immune response in the T and B cell receptors to answer a variety of human immune system questions and other organs as well. We did this in collaboration with Mike Egholm at Roche, who's no longer at Roche, and the idea is, and it should be probably obvious here, is that the human genome, or at least most of them, have 46 B regions, 23 Ds, and 6 Js, and you can recombine in almost every possible combination, producing 2 million recombinants. But in addition to that 2 million, which is already a big number, you have these N regions, which are template-independent, terminal transferase mediated, and they basically can greatly expand and insert long stretches of oligonucleotide that doesn't exist anywhere in the genome and doesn't exist anywhere in the biosphere, in fact. And I'll show you some examples in just a moment. So we're doing this in the context of a time series vaccine experiment, normal adult vaccines in Personal Genome Project volunteers, mainly hepatitis and seasonal influenza vaccines. And what you expect is that you have trace amounts in blue of B cells responsive to any particular antigen that they've never seen before, and then after vaccination or infection, you get maybe 1,000-fold response in abundance of that, of cells reactive to that antigen, and then with a booster or re-exposure, you get additional response, maybe another 1,000-fold. And so we wanted to see this in a time series and analyze it, as you'll see in a moment. This is Yuri Laserson and Francois Vignon and Ido Bachelet, and this is showing that N region I was talking about earlier, where you have the CDR3, which is one of the major hypervariable regions, flanked by V and J, and in the middle is D, which can shrink down to 0. You can basically eliminate the D region through this during the recombination process, or you can expand it with terminal transferase to get as large as 140. So you have this range of 0 to 140 base pairs that's mostly de novo. And this is a logarithmic y-axis, and you can see this interesting distribution with these spikes, and these spikes are because there's already, even before the system decides what antigens are stimulating it, there's selection for full-length, in-frame recombination. So only 1 in 3 of the recombinations produces a full-length heavy chain in this case, and so 2 out of 3, so that 1 out of 3 is highly enriched by a log or 2. And in dark blue is actually all of the data that existed prior to this project in the database, and in light blue is just a few time points from one individual from these time series to give you some idea of the awesome power of next generation sequencing. So this is one of the many computational tools that we employ on this, where we're looking at a time series over a 3-year period for this individual, where we've reset the 0 point is one of the vaccination days going, and here's 14 days before, and here's actually 1 hour afterwards, and 3 days, and 7 to 21 days is where we expect and find antibodies which seem to be responsive to the antigen, in this case influenza. And in this case, because it's high-throughput sequencing, we didn't actually possess these clones. We had to take the sequence and then use high-throughput synthesis to synthesize these antibodies and then do binding assays. So that's examples of semi-epigenomic phenomenon. This is the immune response to the environment. Another critical component of the epigenome is your RNA and chromatin. And in distinction from your genomic DNA, which you can get from any cell in your body, these epigenetic measurements have to be done from the correct cell, and if you want to be comprehensive, they have to be done from every cell in your body. And needless to say, doing every cell in your body is not particularly scalable or embraceable by the volunteers. So instead, what we do is our best effort so far, as maybe something that may turn out to be quite useful, is we established induced pluripotent stem cells for each of the volunteers, and we've published quite a few papers on these particular stem cell lines. And here's some examples of differentiation in teratomas to all the major lineages, including here respiratory epithelium, bone, neuroectoderm, beating cardiac tissue, and in this case, hepatic tissue, which we did in collaboration with Ian Wilmot and George Daly. And you can see not only histological and RNA experiments, but protein level variation from individual variation that may or may not be predictable from the genome by itself, and even activity levels for the cytochrome P450s were discussed in this in one of these papers. To take this to the next level, we'd really like to be doing in situ analysis where we can distinguish the two alleles, or at least quantitate the overall RNA level as you would in RNA sequencing. RNA sequencing is a good quantitative method that allows you to look at alternative splicing and allele-specific expression and so forth for all the genes that are expressed in a given cell sample. And in situ has the awesome power of being able to look at individual cells in their context, making complicated cellular contacts, and indeed making interesting subcellular components, subcellular localization of transcripts and proteins. So as a precedent for this is work that we did now almost 5 years ago, where we did in situ sequencing of what you could consider a very small subcellular component, which is the chromosomes. And the original rationale was for doing haplotyping. Here, you can do haplotyping if you have a trio, but there are many cases where you don't necessarily have the trio, or where the haplotype can get scrambled by rearrangements. In this case, we validated it using carefully chosen homozygous SNPs all along the father, the mother, and the son. And we can actually phase without needing the trio based on single molecule dilution. We can phase the tip to tip the entire 153 megabases in this, doing multiple rounds of local sequencing for short reads, in this case one base pair, where you can see a T at one end of the chromosome and a T at the other end of the chromosome tend to come together due to inheritance from the mother, or a C and a C is the other haplotype you see coming in from the father. Now this is an example of in situ sequencing of sorts intended for haplotyping, but we would like to, and we are moving towards applying this to RNAs, which are present at higher abundance than the chromosomal DNA. And here's an example from some work that we did that John Ock and Chris McCall did with Norbert Perlon, where we're only looking at a few. It's kind of as typical in situs. We're only doing a few, 3 different molecules with 3 different fluorophores at a time, maybe 4 or 5 or so possible. But if you treat this as a next generation sequencing experiment where you can do it over and over, you can see where we're going with this in situ. In this particular case, we were showing the power of using in situs with computational methods to be able to automatically analyze up to over 140 different morphological measures with computational analysis. And I just want to end on, generalizing this issue of imaging as a phenotype that we need in these large cohorts that are intended to be highly integrative, where we have multiple data types openly available for people to develop standards and to develop integrative tools. So here's a completely different level of imaging. Randy Bruckner had a grant to do magnetic resonance imaging variation, looking for variation in human populations, and looking at things like laterality and age, sex, and genetic variation. And so he took advantage of the PGP cohort and just added this as another trait to our list, and he got the benefit of the research subjects. And you can see here are some variations that you see from this PNAS paper due to age and sex, and they're just beginning to figure out some interesting genetic correlates. Now, Ken Nakayama similarly had an interest in behavioral and cognitive tests. So Randy not only measures the MRI, but also behavior and cognitive tests and the genetics. And Ken is interested in prosopagnosia, which is something that affects up to about 5% of the population has difficulty recognizing human faces. They can recognize almost everything else, very intelligent and so forth, but this particular issue. And he's interested in behavioral and cognitive tests in general. And he has a very addictive website called testmybrain.org, which I think now more than 50,000 people have tested their brain. And generalizing this even further, the idea is not to be having one cohort for one specific trait that might interact with a whole variety of other traits and environmental components, is to really try to make it very low cost and high quality, a larger cohort where you've invested enough into it that each additional trait that you get is building on top of all the other ones you have, and you can recontact this cohort. And these people are amazingly enthusiastic, at least so far. And just to give you a feeling for one other way of looking at it is we can look at it as diseases here in brackets, or treatments, which can be either medical devices or drugs or over-the-counter treatments and so forth. And it's kind of here organized by geographic location along the body. This particular person is not a PGP volunteer. He was actually a medical volunteer. He did get electroencephalogram during his lifetime and then donated his brain for anatomical research afterwards. But you get the idea, is that we're trying to engage the community of phenotype collection. This is not just us. This is to feel that they're enabled in some way by having a cohort that they can call upon, and they don't have to necessarily do the genome sequence. Or if they see if someone sees a phenotype that they are interested in, it's already on the website that hasn't been sequenced, then they can target that for priority sequencing. So you've already seen this. I just wanted to use this as a summary slide and open for questions. We're not going from genome to trait, but we consider reducing the cost of collecting all these other environmental components, some of which are genomic themselves, or have in some other way being increasingly well captured by sometimes people just doing more and more monitoring of themselves. It could be the next application of consumer electronics, is self-monitoring. And we're trying to capture this flow. More and more people are coming to us who have already done their genome sequence on their own dime, and so this could be a very cost-effective cohort and project, we hope. So period, full stop. Happy to answer questions.
Hi, George.
Yes.
This is Bert Gold, NCI.
Yeah.
I'm really glad that you mentioned the sickle cell trait case, the sort of high-altitude sickness report from the 1970s, I think it was, in passing, because it kind of provides in capsuo the real problem of phenotyping. I think it's now the year 2010. It's like 40 years or so after that New England Journal of Medicine report. And I don't think we know a lot more about the phenotype of sickle cell trait than from that 40-year-old report in people. And yet the Army, as you pointed out, or the military, wants to know about that trait. So can you give us some of your insight about maybe even how we can know more about that trait and modifier genes?
Well, let me generalize that a little bit. The question is, what hope is there for expanding on a variety of traits? I mean, really, I think the next big push that we all have to focus on is on traits, because the genetics is getting quite inexpensive. I don't mean to understate it. And our ability to get highly predictive, highly medically actionable alleles is really dependent upon breaking up these buckets of these very large buckets that contain, a very large trait like death or cancer or, something that's too large, and breaking up into smaller ones, and then you can actually find the causative alleles effectively. That said, so there are two things I think are big breakthroughs. One is this decreasing costs of devices that you can put on your body or put in your environment and so forth that will help us measure. And probably there will be increasing adoption of this. As with cell phones, initially it will start with wealthy people and work its way down. And the second thing is an increased willingness of physicians to run diagnostics which have multiple modalities in them. I think there was a great fear of that until recently, and it's starting to dissolve, and we'll just have to wait and see to what extent. So it's the individual taking charge and also various clinical resources collecting more data. And then the issue becomes sharing. Because if this is shared very freely, it goes a little faster than if it's put into silos that are proprietary or otherwise inaccessible to creative individuals. Yeah.
All right, if there's no further questions, don't forget there's a reception after this talk at the atrium of this building. And join me in thanking Dr. Church for his talk.